Hi folks, today we're going to talk about genetic disorders and testing, specifically what kind of genetic disorders there are and how do we categorize them and how do we test and screen for them, say when you're having a child and you want to know whether or not this child is going to inherit a genetic disease that you have. Now, first, what exactly is a genetic disorder? Well, it's very simply an abnormality in an individual's genetic material. You know that the genes in your body are instructions to make particular proteins. Well, if we can't make a proper protein, then that genetic change will cause us to have an abnormal protein or a missing protein in our body. Now, in this course, we're going to consider 4 different types of genetic disorders: single gene disorders, multifactorial disorders, chromosomal disorders, and mitochondrial disorders. Really, these are pretty simply understood based on the titles, but we'll talk about each of these individually in just a moment here. But it is important to note that both environmental and genetic factors can play a role in the development of a genetic disease, meaning you can get a genetic disease based on random mutations throughout your life, or even environmental mutations, say from smoking, the carcinogens that are found in tobacco smoke or vaping. Can cause these genetic disorders to occur instead of just inheriting them from your parents like normal genetic diseases you would think of. Now, a single gene disorder is going to be a disorder that is caused by a mutation at exactly one gene. We're only changing one specific gene in the body, and therefore this one gene that codes for one particular protein is the root cause of that protein not being able to be made. Because that segment of DNA, that gene, carries instructions for that production of that protein. So the disease is just the lack of that protein or the abnormality of that protein no longer able to do its job. We know that a protein has to have a specific structure to do its job. Well, what if I had a gene that told it to make a different structure? Then it can't do its job properly. Now, we have different kinds of single gene disorders because they come in different recognizable patterns. We know that genes can be autosomal dominant, meaning they are carried on any of the autosomes, the chromosomes that are non-sex linked and not determined or gender in the forefront. And we can have a dominant or recessive pattern there, meaning that this gene could be expressed or not expressed unless it has 2 copies available to it. Additionally, we might have sex-linked gene disorders, meaning that the individual chromosomes that have the sex genes on them, well, they have other genes on them as well, so those individual genes could be changed in order to have a disorder occur, such as colorblindness, for example. That is a sex-linked genetic disorder because it's on the sex chromosome. Now, a multifactorial disorder is going to be caused by the combination of environmental factors and mutations on multiple genes, meaning that not just one gene is changed, but many, many genes have been changed, and some of them caused by environmental things like UV radiation, carcinogens, etc., etc. For example, heart disease is a multifactorial genetic disorder. We know that it actually has some ties to genetics because individuals that are born from families that have heart disease are more likely to also get heart disease. So there must be something genetically involved in that process there. It doesn't mean that you automatically get heart disease because your parents have heart disease, and that's the key distinction between a single gene and a multifactorial disorder. We can also differentiate that for cancers, specifically breast cancer, have different developments that have been found on multiple chromosomes. So we can consider that the breast cancer itself is a multifactorial genetic disorder of many different genes in the different chromosomes that we have. Most of these are going to be chronic illnesses, and in fact, most common illnesses, such as heart disease, lung disease, have some sort of genetic makeup to it. So most diseases are, unless they're infectious in some way, multifactorial, because there's multiple factors that go along with it, not just environmental choices or lifestyle choices, but also the genetics involved. Chromosomal disorders are going to be related to specifically the long chains of genes that we have that are arranged in what we call chromosomes. And we have 46 chromosomes in our body. 44 of them are what are called autosomes. These are the normal chromosomes that do not have a differentiation between sex. And then we have 2 sex chromosomes inside you. If you're a male, you have an XY, and if you're a female, you have XX. Now, because these chromosomes carry the genetic information, they literally are bundles of genes wrapped around each other. It tends to occur when you have these gametes made, the sperm and eggs being produced inside your body, that problems are going to arise when there are extra copies or missing copies of these chromosomes or parts of these chromosomes missing, or even breaks and deletions and rejoinings of these chromosomes. We have a system to keep these chromosomes in as good a shape as possible, but it's literally like a ball of yarn inside of a cell, and if you tangle that ball of yarn too much, you're going to have problems. And so we might actually look at karyotypes as a picture of paired chromosomes to be able to determine whether or not an individual has a specific chromosomal disorder, such as Down syndrome or Tay-Sachs disease. These are always going to be pretty obvious to look at because there will always be like an extra chromosome or a missing chunk of a chromosome or an entire missing chromosome, and that would be the cause of the chromosomal disorder. Finally, we can have mitochondrial disorders involved, where we have those itty-bitty powerhouses of each and every cell. Well, you actually have genetic information inside the mitochondria. So those things that convert glucose into energy also have DNA. And specifically, they only contain DNA from the mother. The father does not pass down any mitochondria into it because actually your egg cells will have many, many more mitochondria to help the fetus start to develop once the sperm cell is activated inside. The sperm cell does not provide any mitochondria to the fetus. This is a non-chromosomal disorder because the DNA inside of mitochondria is very, very small. Not very many genes are actually processed, and they're not bundled up into chromosomes, so we don't actually count them in a karyotype at all. So those are the different types of genetic disorders. Now let's see exactly how we can test or screen for these different genetic disorders. And one thing we can do is called a carrier screening. A carrier screening is going to determine whether or not an individual carries a copy of an altered gene, whether or not you are a carrier of that copy gene. Specifically, we're going to look for recessive diseases because if you actually have the disease when it's a dominant disease, then you're automatically going to show that phenotype, and therefore you're going to show the characteristics of that disease. A recessive disease means that that particular gene may be on one of the chromosomes but is masked by the other one, and therefore will not show as a phenotype. A carrier screening is often used if a particular disease is common in a couple's ethnic background or if there's a family history of the disease. For example, if you have individuals with a family history of sickle cell disease, well, it's pretty clear that that's going to be passed down through generations on a recessive gene. So even if you don't have that disease, your children might end up having that disease if you have a family history for it. So it's a good idea to get that carrier screening done. Another screening we might do is called pre-implantation genetic diagnosis, or PGD. This will be used specifically for individuals going through in vitro fertilization, where you take the egg and sperm out of the mother and father, and in lab you combine them based on the effectiveness of that sperm to get into the egg as quickly as possible through implantation. And in this process, I'm actually going to have the embryo developed, let it divide a couple times, and then remove a single cell from it to look for those chromosomal abnormalities or genetic changes. And because of that, I can actually make a whole bunch of embryos before implanting them into the mother, and I can have the parent or doctor decide on which embryos to implant based on the success of those potential embryos. And so here's the process of a pre-implantation genetic diagnosis. We have the sperm and egg removed from the mother and father, and once I combine them, they start to make an embryo, what we call a blastocyst, a couple of cells that are continuously dividing over and over. Then I will remove one or two of the cells for testing so that I can see the DNA inside them. And if there is a genetic disorder detected, I'll just not choose to implant that embryo. But if there is no genetic disorder, then I could definitely choose to have that embryo implanted into the mother for growth. But if a couple doesn't go through in vitro fertilization, they can end up doing some screening tests on their fetus before we deliver the child. Specifically, we can do what is called an amniocentesis to see whether or not the child is developing abnormally. We can also insert a needle in there and take a sample of the chorionic villi. The chorion is the tissue that is right next to the placenta where it's connected to the mother, so it's technically part of the child's tissue where they're getting all the nutrients from the mother. And there's little tiny zigzag cells that are there that form a tissue called the villi. So if we take a sample of that, we can actually see the child's genetic makeup based on that tissue. And we can also just do regularly scheduled ultrasounds to prepare the parents for monitoring the health throughout the course of the pregnancy as the fetus continues to grow. Finally, let's say you didn't do any of that and you didn't take care of things while you were pregnant. Well, you can actually do a newborn screening when the person is finally delivering the child. We can just go ahead and detect any genetic or metabolic conditions when the child is actually there. We just take a blood sample and be done with it. And these state tests for newborns typically screen anywhere between 4 to 30 genetic or metabolic disorders, and some states actually mandate this to make sure that you are taking care of your child and getting them the help that they need if there is a genetic disorder involved. Because the goal here is for us to identify any affected newborns quickly in order to provide quick treatment and care, because these first days of a newborn's life are pretty critical to the success of their growth and development.